Disc 5.
The discovery of growth factors, proteins that promote cell proliferation and survival, provoked a proliferation of new biotech companies. Among them, both New York-based Regeneron and Synergen, located in Colorado, hoped to find a treatment for ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, the awful degenerative affliction of nerve cells. Their idea was fine in principle, but in practice, there was simply too little known at the time about how nerve growth factors act for these efforts to be anything more than shots in the dark. Trials on 2 groups of ALS patients failed, and the disease remains untreatable today. The experiments did, however, reveal an interesting side effect. Those taking the drugs lost weight. In a twist that illustrates just how serendipitous the biotech business can be, Regeneron is today developing a modified version of its drug as a weight loss therapy. Another initially speculative enterprise that has seen more than its fair share of dashed commercial hopes is monoclonal antibody, MAB, technology. When they were invented in the mid-1970s at the MRC Laboratory of Molecular Biology at Cambridge University by Cesar Milstein and Georges Kohler, MABs were hailed as the silver bullets that would quickly change the face of medicine. Nevertheless, in an oversight that would today be unthinkable, the MRC failed to patent them. Silver bullets they proved not to be, but after decades of disappointment, they are just now coming into their own. Antibodies are molecules produced by the immune system to bind to and identify invading organisms. Derived from a single line of antibody-producing cells, MABs are antibodies programmed to bind to a unique target. They can be readily produced in mice by injecting animals with the target material, inducing an immune response, and culturing the blood cells from the mouse that produced the MAB. Because MABs can recognize and bind to specific molecules, it was hoped that they could be used with pinpoint accuracy against any number of pernicious intruders, tumor cells, for instance. Such optimism prompted the founding of a slew of MAB-based companies, but they quickly ran into obstacles. Ironically, the most significant of these was the human body's own immune system, which identified the mouse MABs as foreign and duly destroyed them before they could act on their targets. A variety of methods have since been devised to humanize MABs, to replace as much as possible of the mouse antibodies with human components. And the latest generation of MABs represents the biggest growth area in biotech today. Centocor, based near Philadelphia, now owned by Johnson & Johnson, has developed RioPro, an MAB specific to a protein on the surface of platelets, which promote the formation of blood clots. By preventing platelets from sticking together, RioPro reduces the chance of lethal clot formation in patients undergoing angioplasty, for instance. Genentech, never one to lag in the biotech stakes, now markets Herceptin, an MAB that targets certain forms of breast cancer. Immunex in Seattle produces an MAB-based drug called Enbrel, which fights rheumatoid arthritis, a condition associated with the presence of excessive amounts of a particular protein, tumor necrosis factor, TNF, involved in regulating the immune system. Enbrel works by capturing the excess TNF molecules, preventing them from provoking an immune reaction against the tissue in our joints. Still other biotech companies are interested in cloning genes whose protein products are potential targets for new pharmaceuticals. Among the most eagerly sought are the genes for proteins usually found on cell surfaces that serve as receptors for neurotransmitters, hormones, and growth factors. It is through such chemical messengers that the human body coordinates the actions of any individual cell with the actions of trillions of others. Drugs developed blindly in the past, through trial and error, have recently been found to operate by affecting these receptors. And that same new molecular understanding has also explained why so many of these drugs have side effects. Receptors often belong to large families of similar proteins. A drug may indeed effectively target a receptor relevant to the disease in question, but it also may wind up inadvertently targeting similar receptors, thus producing side effects. Intelligent drug design should permit more specific targeting of the receptors so that only the relevant one is blocked. However, as with MABs, what seems a great idea on paper is too often hard to apply in practice, and even harder to make big bucks from. This depressing lesson was learned by Cibia, a San Diego startup associated with the Salk Institute. The discovery of membrane receptors for the neurotransmitter nicotinic acid promised a breakthrough treatment for Parkinson's disease. But as so often in biotech, a good idea was only the beginning of a long scientific process. Ultimately, after giving promising results in monkeys, Cibia's drug candidate failed in humans. Like the unexpected weight loss associated with Regeneron's nerve growth factor, breakthroughs in this area, too, are often born of pure luck rather than the scientific calculus of rational drug design. In 1991, for instance, a Seattle-based company, ICOS, led by George Rathman of Amgen fame, was working with a class of enzymes called phosphodiesterase, which degrade cell signaling molecules. Their quarry was new drugs to lower blood pressure. But one of their test drugs had a surprising side effect. They had stumbled onto a Viagra-like therapy for erectile dysfunction, which may well yield a bigger jackpot than any they previously dreamed of. The market for easier erections notwithstanding, the search for cancer therapies has, not surprisingly, become the single greatest driving force for the biotech industry. The classic cell-killing approach to attacking cancer using radiation or chemotherapy invariably also kills healthy normal cells, typically with dreadful side effects. With developing DNA methodologies, researchers are finally closing in on drugs that can target only those key proteins, many of them growth factors and their receptors on the cell surface, that promote cancer cell